Yeah, good morning, and thank you very much for the invitation to come here. I really like this continent. I'm here every year, and I like to collaborate with Australians. I'm situated at the University of Copenhagen in Denmark. And I'll talk about synthetic plant biology, and I think that's the ultimate way to go green. I'm head of a Center for Synthetic Biology, where we use the modules of nature to build new systems. But I'm also head of a center called Center for Plant Plasticity. How do we develop the robust plants for the future, which are developed to cope with climate changes? And actually, there are some very interesting interactions between those two centers, which I will talk about. Then I've also been head of the Carlsberg Laboratory in Copenhagen. This needs to be turned on. So we get funding from many different sources. We have the Plant Plasticity Center, that's from the Velux Foundation. You may know that company, they're providing sliding doors. Then the government of Denmark has 4 programs, of which I got 1, and that's in synthetic biology. Then I have an ERC Advanced grant, and we have collaboration with Carlsberg, making barley varieties for the future. That's also done with Australians in Melbourne. Then we have collaboration with the company Evolva. There we are producing vanillin made in yeast, the main aroma compound of vanilla. And then we have collaboration with Christian Hansen. With that company, we are making carmine, the red pigment you find in lipstick and red spirits and so on. So we have a large group of projects, and I will talk about one of those today. So what our dream is, 10 years from now, you walk in a park and you have these glass fiber structures. They are solar driven, and in these sculptures, you make medicinal compounds, low-calorie natural sweeteners, pigments like carmine, I told you, flavors like vanillin, aroma compounds, or compounds which make you feel well. So this is a production system we want to get into. And people talk a lot about having a bio-based society, but how do we get there? And that's where our research, I think, can have a contribution. This will involve third-generation genetic engineering. That means good use of genetic engineering. It's just a haphazard uses of expressing one gene and trying to see what happened. Trying to find out how are the modules of nature functioning, how can you build these, also enabling genetic engineering to be used in a good way. There will be a lot of disruptive technologies coming out of this, so that's why one needs to be aware of these developments. We can virtually harvest nature to find out how nature is splendid. We don't need very many samples from nature. We're not using a lot of land to exploit to the benefit of people these competences in nature. It gives a lot of other patenting ideas which might be good to change the patenting system. We need to get this done right, and it's very good to have young people who are very sort of engaged, but we also need to keep them on track so they don't become too big capitalists. So to do this right, and we only work with contained production system when we use genetic engineering. We use our work also to visualize climate change. That's by work in Brisbane together with Phil Hugenholz, where we are studying the microbiome of koalas and the relationship to eucalypts according to climate change. And Australia has an awful lot of opportunities with your special environment and special fauna and flora. So if you want to do sort of use biorenewable energy sources, solar energy is really the prime candidate to exploit or use. And there's 10,000 units as much of solar energy hitting the planet per second as human mankind is actually using. So if we want to use solar energy now, mankind can make solar cells, you can make heat, or you can make electricity. Photosynthetic organisms are a lot smarter. They can use that solar energy to make foods and feeds. You can have drugs from medicinal plants. We can have fine chemicals isolated from plants, pigments, colorants, condiments, and so on. We can make biofuels. That's done by humans. And then we can use the nanomachinery of plants to make more advanced things. So here you have fixation of carbon. Here you have electricity or heat. So the biological systems are a lot clever, much more clever. But we know how these nanomachines are working, and that's what we can use. What's our definition of synthetic biology? That's to engineer biology, to produce biological components or systems, cells or organisms that address the need of society. So these modules of nature are combined in new ways, so we build system or construct system fulfilling the needs of society. And when we do that, we should know what we're doing, so we work with nature instead of against nature. That's a key principle here to get this to work. To share your parts idea, bottom up, to make technologies available generally, but you can patent special uses of the technology. And to create artificial life is not within this scope. And I don't use the word create, we construct, because we are not playing gods. And you should discuss the social and ethical aspects of this upfront. So you explain and get advice from everyone what to do and how to perform. So when I talk about modules, you know that from the electronic industry, you know all these modules, resistors, power plants, power units, and so on. And depending on how you combine these modules, you can build a computer or a refrigerator, a radio, or whatever. You can put them together and build a racing car, and that normally takes a person, typically a man, from one place and back again without any special purposes. But you can also build a bus, and that's exactly the same modules. And that's exactly how nature is using modules in different ways. And a bus takes you to a place you want to go, and you have a time to talk together. So you might end up using that bus for something useful and getting some useful things done. So that's how we should use synthetic biology in our work. And we should get away from this system, which is petrochemically based, into a system which is green, where you use renewable energy sources, and here I'm talking about solar light. So this is the production system we envision, where you have plastic bags, 30 liter volumes each, and they're mounted in greenhouses, and the cells they are producing the molecules you want to make, and the energy input is CO2 from a power combustion plant and solar energy from the sun. This painting illustrates how many people would like production to happen, and it's a very nice painting. Someone managed to bring a sofa there and a naked woman, and there's an indigenous people standing there playing the flute, and the lions are standing there like protecting instead of chewing up the lady. All the plants are moving their flower heads against you to look nice, and there's not a single insect which has made any sort of holes in any of the leaves. This is a very romantic picture, and the people who live there, they know a lot about nature, and we could learn a lot from them. But not many people can live there because all the plants are toxic. So we need to move around, and you cannot feed very many people. Already 450 years ago, this painting was made. You see, you have the monoculture, you can feed a lot of people, but you lose biodiversity. So we need to make a bridge between these two systems in the way to go ahead. Van Gogh was very fond of nature, and he was thinking about how can you use paintings to inspire people to behave right. He called the sun the good God sun, because this was the basis for life. When he was making a painting, he would draw the systems where he saw them. He would never move a house or anything else. And that was his way to pay respect for nature. But he would change the colors depending on his mood. He invited Gauguin and Bernard to his house in France to see how you can develop painting for the future. And Gauguin brought this painting here called The Blue Tree Trunks. And Van Gogh got very upset because these tree trunks were not there. There was positioned there, and he was inspired by the Japanese wood prints. So they had a big argument, and they actually left, and Gauguin got very distressed, and he cut off half of his right ear as a result of this discussion. When Gauguin learned about this back in Paris, he wrote a small booklet called Notes Synthétiques, how you can synthesize natures by modules. So if you use the modules right, you get the right composition. That's the first very nice description of synthetic biology, the principles of nature. There's an artist who has, and she has made now rebuilt Van Gogh's ear. That's in a museum in Karlsruhe, based on his brother's, Theo's great-great-grandson, and now you can build Van Gogh's ear. And that's another way of using synthetic biology. And here you can see the dilemma. How do you want to use synthetic biology? You can illustrate things, but you can also do things wrong. I think there's a trend now with young people. They really want to have a different society. And I've been many decades in science, and there's really a change after the financial crisis. So BioCures is a biohacker place in Sunnyvale in California, and they're making PCR instruments so you can multiply genes, selling them for $400 instead of you pay $8,000 when you buy them from a company. So that's democratizing science. Eric Gentry was, by Obama, was one of the champions of change because she's in charge of BioCurious, and she's getting people to understand what's going on and to discuss what should go on. We have biohacker spaces. It's important for researchers to go into those hacker spaces and be advisors so they don't get things too wrong. There are good people in those hacker spaces, and there are bad people in those hacker spaces. Then I think as a scientist, we have a big, big obligation to perform. If we don't perform, then we are taken over by all these consultants, and then everything goes wrong. So really, I mean, we are the players, and we should play, but if we don't perform, then you are bad players, and that's not good. So there's a lot of responsibility to get these changes implemented in the right way. If you look at the future, whereas the businesses, that's on specialty molecules and fine chemicals. Biofuels, from the beginning, if you did the calculations, you can see it's very difficult to compete on the financial scale. So what to produce? It should be economically feasible. We should not compete with organic chemical synthesis based on petrochemicals. As long as there's not huge taxes, it's very difficult to compete using biological production systems. We should do things which is of benefit to society, environmentally or otherwise not accessible. That could be making drugs which are very costly, which you cannot support by the public health system. It should also be wanted. It doesn't help to produce something and then people don't want it. So the production method should be acceptable, and technology and the product by itself should be acceptable. And don't make yourself dependent on political subsidies. The politicians, they say short term, and before you get your work done, then you have a new politician which you need to accommodate, and so on. And of course, you should make a difference because the science should be good. So I said in the beginning, work with nature. And what's important is to make high-value compounds. We should make something where nature can provide a unifying starting material. So we're not short of the starting material in a natural system. It should be available in high amounts, so you can make a lot of your molecules. It should be driven by light. That means in terms of biochemistry, photosynthetic electron transport is the energy donor using light as the energy source. You should be able to make many compounds. It should be interesting from a science base. This is actually our business plan. You know, when you have a lawn and you put a stone, then the grass becomes yellow. When you move the stone, the day after, the grass is green again. That means there's a huge capacity to build this green molecule. This green molecule has GDPP as a component. It's a chlorophyll molecule. This can be made very easily. This is a cyanobacteria, and if you feed it with nitrogen, the day after, it's green. So the photosynthetic systems can shift the ability to make these starting materials, and this is what we use in our approach here. So I said make high-value compounds. That should be complex molecules which is difficult to chemically synthesize. And there we focused on diterpenoids. I'll get back to those. Then there should be a common starting material. That's the side chain of the chlorophyll molecule, the green pigment of plants. This is made, and if you use it in the chloroplast, then the plant just makes more because it needs to adapt to changing light conditions. It should be available in reasonable amounts. Chlorophyll is made in huge amounts. You should use the chloroplast as a powerhouse because then you have solar energy as the energy source. It should be driven by solar energy, and the outcome, the currency in solar energy is NADPH. So you should find processes which need NADPH to be driven. So again, make this link. You have the output from one process should be the input for the next one. And then you should make desired products. That could be medicinal compounds, food ingredients, wellness compounds, and so on. And we focus on diterpenoids. There are more than 8,000 of these known in nature. The energy system, donor system, solar energy, and it's a chloroplast module. And we know how that sort of part of that module is functioning. We have worked with that since the 70s. We know the output is NADPH, that's the currency. And you need to get that into the sort of systems making these complex molecules. You see the input here is NADPH. There's an enzyme feeding that into this complex here, and that makes the complex chemistry. These processes are separated in the plant cell. This is the chloroplast where you have the energy produced. And the plant decides whether it uses the energy to grow, make seeds or tubers, or make defense compounds. So these processes are separated so the plant can control how to use the energy. What we want to do is to put them together, move these processes here, over here, because then you get more of these products made. Then the plants can't regulate how to use the photosynthetic energy. Illustrated in a non-scientific way, you have the production unit here, and you have the landscape here where the chemistry is made. You have Fred, we call him Fred because it's fluorescence energy transfer, standing here. He's charged with energy, ready to get going. We have Nana, she is a nano machine standing there. And we want to bring them together, because if you bring them together, then she is charged with energy, and then you can make a lot of these molecules. So this is the basic principle of what we are doing using genetic engineering. And the systems we hope is to put these processes together in the same organism or in one single complex. And these are the molecules we're talking about making. Forskolin is selling today in 100% pure form. It costs 95,000 euros per kilo. Ingenol angulate costs 300,000 euros per kilo. So you don't need to make much to have a business. These are the photosynthetic bioreactors we're using. They are 750 liter content. And we're just starting making Taxol. Taxol you can actually chemically synthesize analogs which can use to treat cancer. But this is the most complicated diterpenoids we know of. And we want to make it just to show that we can make any of these molecules where a company would be interested. Today, you can, in the beginning, you isolated this compound from the bark of the yew tree. From 6 trees, which are 100 years old, you can isolate enough of this compound to treat 1 cancer patient with 1 injection. That's why, so it's not made in very huge amounts, and that's why it's so costly. To do these complex chemical reactions, you need an enzyme class called cytochrome P450s. And in the diterpenoid system, you have this side chain of the chlorophyll, that's the starting molecule I mentioned. 2 enzymes converts this into these complex molecules. That's where the organic chemistry gets lost. These are around 40-step reactions if you want to do that by chemistry. Then you have the cytochrome P450s. They decorate these molecules, so you get all these oxygen atoms introduced, and this is really tough. A plant could have 450 different cytochrome P450s, and we need to find those 5 or 6 which does the job. We're making these molecules here, Forskolin, and I'll get back to that, Ingenol angulate, and then lots of other molecules going from cancer to stabilize perfume on the skin of people, and triptolide, which is to sterilize reversible, to make a reversible, it's a reversible male contraceptive. How do we do this? You find the plant which makes the compound, you find out which tissue is done it, then you isolate all the genes expressed in this tissue. You look at bioinformatics, you try to put the gene products together, see how you can build a system in yeast or in a photosynthetic cell which will do that job. And I just illustrate how we do with Forskolin. Forskolin is it's a cyclic AMP booster, so you get a lot of energy when you take it. It's used in treatment of glaucoma in your eye. It's also in testing for cancer and so on. It's produced in this plant, Coleus forskolii. And Coleus plants doesn't produce that everywhere. These compounds are made in specific cell types. And you can, if you know what these cell types are, you can cut them out with a laser, and then you can start with a simple system to see that's where the genes expressed are made, and that's where you can go about to find out how it's done. So in the plant here, instead of extracting all those tubers here, you find out that it's the outer 10, 15 cell layers of the small tubers, which each have a droplet of Forskolin, and that's where the synthesis takes place of this compound, and it's the defense molecules against rodents. So then comes in a lot of analytical chemistry and biochemistry. You can isolate those droplets, and you can see what are the components there, so you get an idea. This could be a good starting material. You see it mimics the final products here. And then you express those terpenoid synthases, and you find out a good combination here actually give you this starting material. And then comes the challenge to find those cytochrome P450s, which decorate the molecules to give the final product. And again, you do LCMS technology, and I will not go into the details, but you get the first compound converted into this one, you get side products, and you can look at the masses, and you can see you get oxygen atoms introduced. Then you introduce a second P450, and you get more products, and you get sort of structures which starts to mimic the final product. And then you introduce the last enzyme, which is an acetyl transferase, and you get your product. And the beauty here is that suddenly you only get one product. That's because now you're mimicking the way nature is doing. So nature will make a huge set of these molecules with different enzyme systems. but each one is very specific. So it makes a metabolite and an enzyme complex. And if you can rebuild this, you can do something, you can exploit the beauty of nature in this production system. So this is how forskolin looks now when it's produced in yeast. And that means, of course, that isolation is a lot simpler when you don't have 200 other molecules you need to separate it from. When you get these modules, the terpenoid synthases, you can take the first one from one plant and combine it with other modules from other plants, and then you can make completely different core structures, also compounds which are new to nature, or at least not known in nature yet. And many of these will have bioactive uses. This is not simple research, and some of the complex equipment and also costly equipment is shown here. But it's feasible. We sorted out the forskolin pathway is 5 man-years to do that entire pathway. So still costly, and you need a lot of infrastructure, and that's why collaboration in this area is very, very important, but it's feasible. And these are the algae bags we want to produce. I really like working with Australians because you have a lot of opportunities here. You have many different challenges which are tough to sort of face, but you also have opportunities. You have a unique plant system, and we, together with people in Western Australia, here in Adelaide with Susan Semple, and people in Melbourne, we now applied to the Novo Nordisk Foundation in Denmark to work with the Eremophila species. These drought-tolerant species can resist high solar energy, drought. They need to defend themselves. They need to spend very little energy to all these processes. So the terpenoids they are making are bound to be really, really important drugs. They have very, very interesting properties. So we are together, we will do that, and we'll try to set up sort of a foundation to do this work. We are in the second step of the Novo Nordisk application. We applied for $3 million over 3 years, and I really hope we get that grant. Another project we're running that's together with Phil Hugenholz in Brisbane, that's on the eucalypt and the koala interrelationships, because I think that's a very good way to illustrate to the general public how climate changes will affect the wildlife sort of feeding behaviors and our whole sort of way of operating. So the koala only feeds on the eucalypt leaves, and they change composition depending on the heat spell. The koala can only metabolize all these toxins present because when the joy is born, the female empties the appendix, and the microbes there is eaten by the joy to get the microorganisms sort of in the digestive tract able to digest all these toxins. So that relationships we are studying. So we are studying the microbiomes of the sternal gland of the male, which he used to communicate, of the gut. So we are doing sequencing of all these microorganisms and assembling their genomes and looking for interesting genes there. That's the sort of the commercial part of it. The koala appears to be able to degrade lignin. And in the biofuel industry, to degrade lignin is actually an important asset. So the biotech company involved will look for that one. Climate change will make a lot of these molecules here with cross-linked proteins, and that makes it difficult for the koala to digest the eucalypt leaves. So we are looking at different eucalypt species and really doing a detailed characterization. And there are many interesting compounds in the sternal glands. In the sternal glands here, they're coming from metabolites in the eucalypts, and they are excreted here, and they are sort of signaling molecules for the koala. So they're very interesting interactions here. The appendix is huge, and the koala have to digest a long time to get energy out, and this is where you have microbes, which will really be very, very interesting. So we want to move from this system here into a green system. When we do the forskolin work, we need very little plant material. You do the enzyme work, the analysis with a 10 millimeter leaf disc. The technology is extremely sensitive, and likewise the genes. So you're not sort of ruining acquiring entire fields. We need to get this presented right, and this is a picture of the little mermaids in the harbor of Copenhagen. And 200 meters away, there's another artist who made sort of what he calls a genetically modified paradise. So this is a genetically modified mermaid. They're both genetically modified with a fishtail and a human body. This is a stone carving from Sweden. I think if a scientist had made this one, that scientist would have been sort of accused for plagiarism because it looks like very much that one. So we need to communicate and get our messages across, and we need to do the right things. And thank you for your attention. It's tough to be in this area because it's interdisciplinary. There's ups and downs. And robust funding and the understanding of politicians about this is key to success because it's really, really tough. You bridge. There are lots of gaps. You don't use model plants and so on. It is tough, but it's worthwhile. Thank you.